for giving this opportunity to talk about my research work. I, I really thank you for starting this series of seminars and bringing out new folks to or all folks to actually to showcase their work. So I am Ruchika Bajaj, and I am trained as a membrane protein biologist, and I will talk about understanding ABC transporters to navigate human diseases. So this is a little snapshot of my scientific journey in ABC transporters. So I did my graduate school at Purdue, where I studied bacterial ABC transporters, and mainly one of the protein was like from FTSX plus PCSP from Streptococcus pneumonia, and MAL-G501 is the binding protein independent mutant of E. coli maltose transporter. And then I came to postdoctoral training at UCS, where I worked on P-glycoprotein and MRP4 mammalian ABC transporters. And this journey was accompanied by lots of mentors, and you can see that I have lots of mentors during my grad school as well as my postdoc. Okay, so I will start from the basic. Like membrane protein actually constitutes like 20 to 30% of proteins encoded by the genome. We are all organisms we are made up of cells, and these cells have membranes, and these membranes are made up of carbohydrates, lipids, and proteins. And proteins embedded in these proteins in the membrane are called as membrane protein, and they could be either embedded in the membrane like transmembrane protein, or they are just associated with the membrane called as peripheral protein. And my major focus has been on the integral membrane proteins, and these membrane proteins could be receptor and taking the signal from the external site and transferring into the internal site. And then they could be enzymatic in nature and catalyze a reaction in the cytosol, or they could be transporter or ion channel and moving the solutes around across the membrane. And the good thing is about ABC transporter is that they can actually fall into all these categories, like receptors as well as enzyme and transporter and ion channels. So now I will give you a short introduction of ABC transporters. ABC transporters are ubiquitous in nature, and they are the large superfamily of like membrane proteins, and they are present in prokaryotes as well as eukaryotes. They are usually made up of two TMD domains, which form the pathway across the membrane, and two NBD domains that's called as nucleotide binding domains. And these nucleotide binding domains have ATP binding sites, and these binding sites are being composed by these conserved sequences from A loop, P loop, ATP, switch histidine, or Walker B, Walker A, and D loop and signature motif. And these ABC transporters are actually, since they're ubiquitous in nature and they're present everywhere, they're expressed in like almost all tissues, and they are involved in lots of physiological processes. And because they are involved in so many physiological processes, they are also involved in diseases. So this is a very important class of membrane proteins, which is significant to medicine. So it is very important to study these transporters and really navigate diseases through it. So here is this is a basic alternating access mechanism in ABC transporters. Usually a solute is being, so these TMD domains move toward, they open to the either side of the membrane and to cross the solute across the membrane. And there are some prokaryotic ABC transporters which also use these binding proteins which have high affinity towards the solute to sequester the solute and then transfer it to the TMD domain. So here I will first briefly introduce you to my research work in the graduate school, and then I will further move towards the postdoctoral work. So in the detail. So I worked with, I started my journey in ABC transporter with with Amy Davidson, who was a pioneer in the field. And then because she died during my graduate school, I also continued my graduate school with Cynthia Stoffaker. And then at the same time, I also collaborated with Michael Winkler. Winkler Lab had shown an interaction between FTSX and PCSP, which are and FTSC, which are like essential protein in Streptococcus pneumonia during the cell division process of this pathogenic bacteria. And we wanted to reconstitute this system into the in vitro, and I kind of challenged the literature, and this FTSX was which a membrane protein, which was found to be toxic to be expressed in the membrane, and found to be toxic in the membrane. And I challenged the literature and expressed it and purified it as a single band on the gel. And after purifying it, I reconstituted into the nanodisk, and showed that it is a dimer by cross-linking experiments. And besides that, I also worked on FTSE, which was an associated protein with it, and purified it, and also showed that there it can bind ATP with a similar KD value as other ABC transporters. With that, I also worked on a PCSB, which was not a membrane protein, but it was an associated protein with the FTSX, and it was known to bind membranes, but it was not characterized well. So this is where I found that it can actually bind the lipids part, and with using the head, like positively head charge of lipids, especially like PE. And this binding or interaction happens via coil domain rather than chap domain. So here are the conclusions that FTS6 is a non-toxic to express, which was known to be toxic in E. coli, and then we considered it as a dimer. And FTSC can bind ATP analogs with similar affinity constant as other ABC transporters. And PCSP can also bind the lipids with positively charged head groups via coil domain. Okay, now I will shift gears to MALG501, which was an in-binding protein independent mutant of E. coli maltose transporter. And so here it had two mutations, isoleucine 154S and isoleucine 135A in the MALG. So maltose, just to describe you that maltose transporter consists of MALG, MALF, and two nucleotide binding domain, and it had specific mutation in MALG part. And this specific transporter fetched my attention for the fact that it had high maltose transport activity, which was showing biphasic behavior with increasing concentration of MBP, and it did not require any MBP in general to go for the basic activity. So here were some questions like, how does it bypass the essential requirement of MBP, and why does MBP play an inhibitory role in its catalytic cycle? So here what we did is we had tools, the EPR, basically we had cysteine mutants which could be studied to use to catalytic cycle of maltose transporter using EPR. So we used the same mutant and had the MALG mutations in that, and we deduced its catalytic cycle, and we found that it kind of exists in an equilibrium between open state and semi-open state, and this activated state has high affinity with MBP, and because of that it can inhibit MBP can actually inhibit it, or if it is not being interacted by MBP, it can just go into its own cycle. So there were two major points, that it exists in the equilibrium between resting state and semi-open state, and there is a high affinity interaction between activated state and the MBP, which is causing this biphasic behavior that explained. Yeah. So during my graduate school, I mainly focused on bacterial ABC transporter and exporters, and now during my postdoctoral training, I focused on mammalian ABC transporters. So just to let you know, there are 49 human ABC transporter, which are grouped into 7 subfamilies. And here are these 7 subfamilies are A to G, A, B, C, D, E, G, A, B, C, D, E, and F, and G. And I mainly focused on ABCB1, the first member of the B family, and ABCC4 or ABCC ABCC4, which is the fourth member of the ABCC family. So I will first talk about my work on MRP4, that's ABCC4, multi-drug resistant 4, and then we will talk about the P-glycoprotein. So like other ABC transporter, it also has the 2 TMD domain and 2 NBD domain, and we do have the structures, and I will talk about those now. So MVP was discovered in 1999 because it was associated with the strain which was really resistant to PMEA. It was really highly expressed in that strain, and that's how it was identified in 1999. And it is actually expressed in different organs: brain and kidney, prostate, liver, and even gastrointestinal tract. And the unusual thing about MRP4 is that it can be expressed on the apical side as well as on the basical side. And this MRP4 is very, very clinically significant molecule for the fact that it is involved in various processes like cardiomyocyte contractility and proliferation, sperm capacitation or cell migration, and cell differentiation and proliferation. And even because it has lots of different diverse substrates, including PGE, endogenous substrates like PGE2 and leukotrienes and CMP and CGMP. So it can be involved in various processes, and as well as there are drugs which actually are also substrates of MRP4. So it can be involved in the drug resistance and other diseases like cancer, cardiac diseases, or liver disease and inflammation processes. Or because it can efflux lots of chemotherapeutic drugs, so it can also lead to chemotherapeutic failure especially in cancer. So these are the reasons actually make this MRP4 as a very clinically significant molecule. And the good thing about this project is that inhibition of MRP4 can actually reverse disease state. So here you can see that MRP4 is usually overexpressed in pulmonary hypertension, and it has been mimicked in the hypoxic state as well in mice models of pulmonary hypertension, where you can see that there is a thickening of these layers, the thickening of your veins or arteries, and that thickening can be reversed to normal by just inhibiting MRP4. So with that, these considering endpoints, so there have been studies which have been looking for MRP4 inhibitors, but they are not actually selective and non-potent. They are not actually selective and potent in nature. And also, we have other functional studies going on for MRP4 where different mutations have been studied for the expression and function. And on top of that, single nucleotide polymorphism has been also associated in MRP4 with various clinical conditions. For example, septic attack, low levels of electroprost in open-angle glaucoma, or high-grade thrombocytopenia, or thyroid toxicity and leukopenia, etc. So these are the various SNPs in MRP4 which have been associated with expression and function. And so to explain, so there's a lot of literature on MRP4 which is actually earning for MRP4 structure for last 21 years. And to understand all this structure-functional relationships and to design new drugs for it. So here there are basic questions like what is the basic architecture of MRP4, and how is the mechanistic cycle, catalytic cycle of MRP4, and where is the substrate binding site, or what is the role of asymmetry in NBDs in catalytic cycle, and why and how missense SNPs can affect the function. So these were the questions which we wanted to learn about. So this is why we wanted to have a biochemical prep of MRP4 and to go for the structure. And it took us so long, like 21 years, to actually get there, for the fact that membrane protein structural biology had been going through so many struggles during my graduate school as well as postdoctoral training. So for example, membrane proteins are very hard to express. They have very low yields. Then we have expensive detergent. And even if we use this detergent to extract from the native membrane, they are just they are in artificial membrane again. So then you have to put them back into nanodisk, and they are so floppy and heterogeneous in nature. And besides that, crystallization is random process, and there are long purification times for the membrane proteins. So, and however, to get beyond or overcome these challenges, we have also got advances in the field. For example, we have got nanodiscs, which can reconstitute into the native environment, and then we do have some amphifolds and novel detergent which can extract it easily. And then besides that, we from 2013 we have got lots of advancements in the electron microscopy, cryo-EM especially, and then advances in the eukaryotic membrane expression systems. And then again we had like computational algorithm which have made like robust system for the cryo-EM purpose. Like we do have like different softwares, Reliant, CryoSpark system, and Serial EM going further now. For making this process, single particle cryo-EM, such a robust and routine process. So after these all advances, I'm happy to say that we can actually get the structure for MRP4, and I will show you that. And these all advances in cryo-EM technology and membrane expression system has also revolutionized the field of ABC transporter as well. So you can see that ABC transporter field had started in 2002, and then the structures with BTUD structure by Doug Rees, and then it had been going in lag phase until 2012. But since 2013, when we got the advancements in cryo-EM and also expression system, then it kind of started moving further along. And then by the last era, like from 2017 to 2021, it had been like blooming, like bursting basically. And the funny thing was that 2020, which was a COVID year, was the most productive year for the ABC transporter field. And yeah, so structures have actually introduced a novel fold classification in ABC transporters. Now, so you can see that there have been different kind of architectures. I mean, here you can see swap, helices, and now we have extracellular domain as well. Here we have binding protein domains. So there are different classification based on structures as well. So I now coming back to MRP4. MRP4 was overexpressed in mammalian and insect cells. We had gone through different orthologs, mouse, rat, and bovine orthologs. And we started with the insect cell expression system, and we could really get a nice band by optimizing different temperature and multiplicity of infection. And we chose bovine MRP4 for the fact that it was almost 90% identical to human MRP4. And then we tried to purify it, and we could purify it as a 150 kilodalton protein. And then we reconstituted into nanodisk, and you can see that we had both bands in the gel. And you can also see that we had particles, characterize those particles by negative stain electron microscopy, and you can see that two NBDs were bulging out from the major nanodisk shield plate. And then afterwards, we also confirmed the integrity of reconstituted bovine MRP4 by using mass spectrometry. And here you can see that all the red color peptides were shown in the data. And here you can see that bovine MRP4, and then we also characterized the functional activity of bovine MRP4, and we could see that there was a basal activity going on in here. And it had going through the Michael-Mentel's curve. And on the other side, E1202Q mutant, which is, sorry. So glutamate is a very important residue in NBD domains, especially because it kind of polarizes the water molecule to catalyze the ATP hydrolysis. So because it is very important, and if it is mutated, then the ATPase activity goes down, and which was actually demonstrated in this experiment as well. So after confirming the functional activity and going through these controls, and also we characterize the ATPase activity with its substrates. So here we found that its substrate PGE1 and PGE2 actually could stimulate its activity from the basal level. And this fold stimulation was more for PGE2 and PGE2 and DHEAS, but not for the other substrate, CMP, CGMP, LTC4 or E217G, which are known in the literature. So after confirming all this expression, purification, reconstitution, and functional characterization of bovine MRP4, we move towards the high resolution cryo-EM structures of bovine MRP4. And here you can see that it's a kind of a long polypeptide chain with the 2 TMD domains of six helices and then 2 NBD domains. And we determined the structure of ABC transporter in 3 different states. The first state was apostate, which had NBD domains very open, open in the sense very far away from each other. And on the others, and you could see that there is a swapping of helices, that TM4 and TM5 are coming from TMD1 to TMD2, and then TM10 and TM11 are coming from TMD2 to TMD1. And then substrate bound, then we also got another structure in substrate bound state here at the 2.7 angstrom structure, and you can see that this substrate is bound in the middle of the bilayer. And ATP bounds, then we also had the ATP bound state where these NBD domains were close together. So basically, so these are far more open in the apo state, which are closing with the when substrate is bound, and then they close it more when ATP is bound. And here, analyzing these structures also gave us more conclusion, and I will explain those now. Before I get into those conclusions, I also want to explain that here are the sequences for NBD1 and NBD2 major sequences like Walker A, Walker B, and signature sequence. So this is the E I was talking about, glutamate, which is very important for the water hydrolysis, polarizing water for the ATP hydrolysis. And you can see that there is a deletion of 13 residues in the NBD domain, which is present in NBD2. And because of this asymmetry, and there is a change of aspartate in the NBD1, which is E in the NBD2. So here you could see, so because of this asymmetry in sequence, we can also see this asymmetry in structure as well, which is being induced by substrate in MRP4. So in resting state, these NBD domains are pretty much open, and they are at the similar distances. However, when it comes to a substrate bound state, now one ATP binding site, which is consensus binding site, which is degenerate binding site, is far away than compared to consensus binding site, which is closer in nature. And on the other side, this asymmetry is again lost when ATP bounds and they come close together to like 5 angstrom state. And this, as I mentioned about this asymmetry, we will also see that this asymmetry is also pursued further at the NBD2 and TMD2 interface, where we have this deleted sequence going on. Where we have this deleted sequence. So here you can see that this region is the where we have this deleted sequence to be superimposed, and you can see that there is a specific interaction between histidine 1111 and glutamate 292. 292, which is strongly held together during this NBD2 and TMD2. But in NBD, there is no sequence like that. So this is more NBD1 and TMD1 interaction is very loose in nature compared to NBD2 and TMD2. And in another conclusion, ATP is bound by conserved sequences in bovine MRP4. So here you can see that the sequences I was talking about, Walker A and Walker B sequences, these are all interacting with the ATP here. You can see that there is a ATP ring which is stacking around this tryptophan in degenerate site, and these all phosphate residues are bound by the lysine, positive lysine, or glutamine and histidine. And similarly, on the consensus site also, that adenine ring of ATP is stacked towards like tyrosine, and there are other K positive residues which are binding to the phosphate residues. And here's the Q 1202, which we used for the structure bound state. That's the EQ mutant, and this is kind of mutated in this state. So after going through this asymmetry and substrate like asymmetry in NBD sequences, we also tried to check out the substrate binding site in MRP4 using LTC4 bound MRP4 structures. So we did sequence analysis, and we took the corresponding residues from bovine MRP4 structure, which were bound to the substrate, which were already known by the Juchen structure, and then we found that there were many residues which could be comparable, and there were few residues which were already known to be interacting with substrate in the literature. And when we found those in the substrate, and we found that many of these residues were actually comparable to what we found in the structure. So here are those structure, and you can see that substrate is kind of stacking in between these hydrophobic tryptophan, and also these hydrophobic residues like phenylalanine, and polar and non-polar residue like leucine. And you could see, so this is a DHEAS bound state where here its oxygens are being bound by water and arginine 946 and histidine. And again, here also sulfate a sulfate group is also bound by water bridges and the positive charge residues like R362 or glutamines 849 or 845, and Y70, Y2728. So this is how the substrate is bound in the MRP4. So here is a small clip video which will show how the R946 actually reorients to bind the substrate. You will see that this comes close to the cavity and while binding it, but while removing it out, then these helices come close together and they kind of excise or pull out the substrate. I can show this video again. You see, so these residues are coming in to interact with your substrate at the substrate binding site, and when the ATP binds, then this kind of pull out this substrate out. So they come to, those helices come close together and the substrate comes out. So here I will show this concept again here in this slide. So you could see that APO is more relaxed or more open in nature than compared to DHEAS, and but in ATP bound state, these residues are coming more close together so that the substrate can exit out. And this here major conformational changes are happening in these helices TM3, TM6, TM9, and TM12. So based on all this information we had from asymmetry as well as substrate binding sites and the movement of helices, we are proposing here like the mechanistic cycle of MRP4, where here MRP4 exists in a resting state when substrate bounds, and it brings this all TM domains together. So that NBD also come together. However, when ATP binds, then these NBD close more to, they come more closer, and find that leads to the exit of the substrate, and then it leads to the ATP hydrolysis and resting and recovering back to the resting state. So here is another video which will show a bit more about conformational changes during catalytic cycle of MRP4. So it's breathing molecule and substrate comes in, which brings TMDs together and surely with NBDs together because they are bound to the TMDs. And then there is ATP coming up that closes the molecule again, and that leads to the exit of the substrate. And while recovering, this ATP hydrolysis happens and ADP is released, and it opens up again back to the resting state. And so besides learning the structure and functional relationships, we are also trying to understand the genetic variants which were also detected by structure. So there are 23 single nucleotide polymorphic variants known in literature which have been characterized for the function as well as expression. And we located on the structure and they classified them into 4 different categories. Like green were pore residues, and then orange are the more surface residues, and then pink are near the interface mutations, and then blue ones are actually near the ATPase sites. And from all this 23, we mainly focused on E75K and K304 and G187 for the fact that they had the high allelic frequency. And you can see that here, if G, which is a glycine like small, without any side chain, is mutated to W, which is a really bulky residue, it can actually cause clashes with the ATP bound and can really cause a significant problem here. Similarly, K309, which is R304 in the bovine, forms a H bond with N301. And if this is mutated to asparagine, it can lead to the loss of hydrogen bond and so can cause a problem in the function of the protein. And besides learning structure, function, and the genetic variants, we are also trying to learn the evolution of this MRP4. And you can see that here we have got these this is in more different, like conserved is the more magenta, which are conserved are more magenta ones, like here, which are in the pore regions and the NBD regions, but most of the residues in the TMD regions are variable in nature, which are cyan in color. And we have here 2 branches of MRPs and 2 branches, and MRP4 is in the lower branch. So conclusion, we have got high resolution CryoEM structures of mammalian MRP4 in nucleotide free state, substrate bound state, and nucleotide bound state. They have been determined, and we have learned about the architecture of MRP4, which is type 4 fold now, and also substrate binding site and the role of substrate in inducing the asymmetry in NBDs, and how is the catalytic cycle of MRP4 and associated conformational changes. And we have also tried to interpret the functional effect of genetic variants in MRP4. And so here is the, we also want to continue this project further by learning MRP4 catalytic cycle using SMFRET so that we can coordinate those conformational changes to more dynamic nature. And then we also want to try to go for the structure guided identification of chemical probes for MRP4. And we also want to extend our genetic variants to the whole database, like nomads, for prioritizing SNPs for the field of precision medicine. And besides that, we also learned that MRP4 actually interacts with the PDZ domains. So it would be nice to understand their interaction with CFTR and PDZ domain, and how does that help in the physiological process. So I would like to thank the whole team. It was such a collaborative project, and we started with Fong and Stroud Lab, with the expression and purification. And then we collaborated with Ifan's lab, with the graduate student in Ifan's lab, and optimized the cryo-EM conditions. And then for mass spectrometry, we took help from Gigi. And then finally, Sergey also helped with the later structures. And this was the whole team of mentors which who was behind us and stepping up, like guiding our steps in the project. And I would also like to thank UCSF facilities with like mass spectrometry and ultramicroscopy, which are running well, uh, and letting us do our projects well. And surely I want to thank NIH PO1 grant, which used to help together this whole team of 15 people who will come together every month and discuss the science together. And I also want to thank one of my postdoctoral fellowship, American Heart Association, which helped me fund it throughout the later phase of this MRP4 project. And so now I have talked about the MRP4, and now I will take a little step ahead and shift gears towards the interaction of commonly used molecular excipient with P-glycoprotein. So here I wanna talk about P-glycoprotein is present on the apical membrane of the intestinal lumen with other ABC transporters like MRP2 and BCRP. And so during the gastric intestinal tract, and because 50 to 60% of drugs are substrates of P-glycoprotein, so it is one of the kind of decider that which drug will be absorbed in and or available to the blood side. And also, so basically this is more of a major player of pharmacokinetics and pharmacodynamics of the drugs. And besides that, it is also present in the brain blood barrier. So there it controls like which drugs will be monitor which drugs will be coming in or coming out. And in chemotherapies also, it kind of fluxes out lots of drugs out. So it is really important to study the interaction of drugs or other excipients with with P-glycoprotein, various compound with P-glycoprotein. So now I will introduce the term excipients. So excipients are inactive ingredients in tablets. So we have tablets where we have in active ingredients, and everything else like colored compounds or solubilizers, etc. Everything else which is being added to the tablet is called as excipient. And they are actually added in the tablets for various functions. For example, they can act as a buffering agent, sweeteners, stimulators, binders, substabilizers, surfactants, or solubilizers, or they can just act as a coloring or flavoring agent. But these functions are very important, and they can also act as antimicrobial preservatives. So they are usually known to be very safe and inactive ingredients. However, there are some times that these excipients can be really interacting with components and can really change the pharmacokinetics of the drug. For example, here, if excipient has no interaction, then we have this blue curve going on. Here, we are measuring the drug concentration in the plasma. And if it is acting as an inhibitor, then it can change the profile to like really high concentrations. And if it is acting as an inducer, then it can lower down the concentration of drugs. So basically, the interaction of these excipients with these protein components, especially with P-glycoprotein, can actually change the pharmacokinetics of the drug or concentration of the drug. So it is very important to understand these interactions. So here we use the screening procedures to screen this whole 123 molecular excipients in the library. And we use two different assays, calcein AM fluorescence-based assay and radioactive-based digoxin flux assay, which I will describe in next few slides. So, and once after the screening through this assay, whatever potential inhibitors we get from this, we go through those IC50 studies or dose response curve studies, and we compare it to the intestinal concentration and see how they can be formulated in the next generation of drugs. So here I will start with the calcein AM assay. So here we had two different cell lines. One was PGP overexpressing cell line, and the other one was calcium was empty vector cell line. And this calcium AM is a hydrophobic molecule which is passively diffused into the cells and being cleaved by esterases to calcein. And if it is if it is if PGP is overexpressing, then it does not get chance to get cleaved by esterase. Instead, it is just being effluxed out, and so there is a less fluorescence in PGP overexpressing cells. However, in the empty vector cells where there is not much of P-glycoprotein and most of the calcein AM is being cleaved by esterases to calcein, there is high fluorescence. So this is the difference between difference of fluorescence in two cell line is measured as PGP mediated transport. And here we confirmed that here you can see that here's the fluorescence. So empty vector fluorescence was threefold higher than the PGP fluorescence with because of the high expression of PGP. And then this fluorescence was increased in both cases, but the increase for PGP cell line was higher than compared to EV cell line. Again, because due to the expression of P-glycoprotein in P-glycoprotein overexpression cells. And so we converted this fold change fluorescence in normalized calcium AM ratio, which was measured by ratio of fluorescence of PGP cell line in presence and absence of the inhibitor divided by the similar ratio of EV cell line. And so this normalized calcium accumulation ratio was 2 in case of verapamil. So we, after validating these assays, we did the whole screening with this assay, and here you could see that we ordered them according to the normalized calcium accumulation ratio. And then we could we also normalized these different ratios so that we can have those only specific inhibitors which had effect on PGP cell line but not on empty vector cell line. And here we took the threshold of 1.4 for seeing the difference between on PGP cell line, but for empty vector we wanted to have negligible effect. So here we wanted to pick up those compounds only when there's negligible effect on PGP cell line, basically no change in fluorescence. And so we picked up on two compounds, DC brown number 1 and DC red number 6. And when we did the dose response curve for these specific compounds, and we found that there was not much inhibition going on. And this is where we were surprised that when we predicted that the fluorescence might be a problem for this colored the fluorescence measurement could be interfering with this color compound. So this is why we moved towards the P-glycoprotein mediated digoxin flux based by radioactive based assays. So we moved toward this radioactive based assay for the fact that it was more sensitive, and also we were trying to mimic the real gut system. For example, here we had two chambers, chambers, apical chambers as well as the basal chamber, and the cell line was actually placed in on this membrane. And then apical chamber was mimicking the gut lumen, and then this intestinal gut lining was our cell line, and then basal cells were mimicking the blood circulation process. And you could see that this cell line was overexpressing human MDR1, and the substrate was present on the basal side, which was moving from the basal to the apical side. And here we validated this assay by saying that MDR1 knockout cell line did not have any digoxin flux, but human MDR cell line had flux, and which was inhibited in the presence of cyclosporin up to the tenfold. So there are two potential in, and so after validating that previous assay, after validating this previous assay here, we screened the whole library of 123 molecular excipients and ordered them with the digoxin flux assay. And here we also try to count the mean permeability of about 3%. So here we took the threshold of 60% for inhibition part for the normalized digoxin flux. And after taking that for 60% part, and also we wanted to make sure that this excipient is not affecting the mono integrity of cellular monolayer. So we picked up on two compounds, light green SF yellowish and beta cyclodextrin. And then we did the IC50 studies, and we found that they were actually inhibitor. So with their IC50 in high micromolecular range, and we compared those with the maximum intestinal concentration as well, and you can see that here that beta cyclodextrin had threefold maximum concentration than IC50, but for light green CF yellowish, it was similar to the IC50. So basically, we want to be careful about our future formulation when we are preparing drugs with these two specific compounds. So here is the conclusion that we have screened this whole library of oral excipients against P-glycoprotein using calcein fluorescence assay and digoxin flux assay. And most of the screen excipients are safe or inert with respect to P-glycoprotein. However, beta cyclodextrin and light green SF yellowish were found to be inhibitory at high micromolecular range in digoxin flux assay. So, and this information can also help generic companies which can develop new drugs at the cheaper rates. And for this project, I would really like to thank Diana, who was mentoring me during the whole study and making sure that my steps are right. And then I would also like to thank Kathy, who helped us in mediating the collaboration with FDA. And Liza was my graduate student who helped me with the dose response curves for the calcein assay. And Ling was in Kathy's lab, and she was a great mentor, and she provided me reagents and also answered my question whenever I had. And again, I would like to thank FDA for providing us the discussion platform every month that we could discuss this data and make sure that we are going through the right steps, and for funding as well. And I would also like to thank American Heart Association for funding my postdoctoral fellowship. With that, I am happy to say that my scientific journey in ABC Transporter has been leading these four systems: FTSEX and PCSB and MALG501 and P-glycoprotein and MRP4. And besides that, I had also worked on another that actually intrigued me to pursue to understand how is the field of ABC transporter is evolving, and we just had a paper on that. And it was because I was mentored by such a nice team of mentors. I thank you all those all those mentors. And then I'm happy to take questions.
Awesome. That was a really cool talk. So I will go ahead and jump into the questions. We got a few here. Let's see. Can you talk? This was back at the beginning, but can you talk more about how you confirm the protein reconstitution by mass spectrometry?
Yeah, so how we confirmed that by mass spectrophotometry was like first you see that here in the gel.
Mm-hmm.
Once we purified it and then reconstituted it like this, we had taken this sample.
Okay.
We had cut this sample and sent it to for the mass spec. And then by mass spec, I mean basically they had done, I mean I had done the process for the entryptic digestion, and then those after trypsin, then we had sent it for the mass spec, and they had detected the peptides, and then we had and those are the peptides basically you have seen in the sequence,
Okay, so
and that's
so
how
so
we
you
identified.
you break the protein down, and you know, okay, if it's what I think it is, it should have these different peptide groups, then and that corresponds to a certain I guess M over Z value. And as long as you have all those peaks, you say, okay, yeah, that's the one.
So the major like how much is the coverage of specific sequence, that's how we confirm the integrity of our protein.
Okay, okay, interesting. That's really cool. I've done a little bit of mass spec, but not not on something nearly this complicated. So that's really fascinating to learn about. Let's see. How fast does the MRP4 catalytic cycle occur?
That's a very good question. So here is the basal activity. You can see that it can hydrolyze. I mean, it depends upon obviously ATP concentration. At the higher
Hmm.
concentration, it can hydrolyze 20 nanomoles per minute and per milligram of your protein.
Okay, that
Yeah.
seems relatively quickly, I think. But I don't have I guess I don't have that good of a feel for it, but it seems pretty fast.
So it says like 20 nanomoles of your ATP will be converted in per minute per
Yeah.
milligram of the protein. Yeah.
Okay. Interesting. Let's see. Can you give an example of an application of the MRP4 work? Will it allow development of new medicine?
Yeah, so now we have got structures and we can actually go towards the structure based drug designing. Like now we have also got the substrate binding sites here, you
Hmm.
could see. And surely we can screen out different libraries which can bind more tightly or with the higher binding scores, and we can try out those in experiments uh, to see if they are real inhibitors, and we can pull those on the specific library. And then there is one another problem going on in the literature is that we have different MRPs in the same family, MRP1 to
Hmm.
MRP7, and most of them are being inhibited by just specific like one inhibitor itself. So we want to design inhibitors specific to different MRPs. So that's
Okay.
where I think structural information can help.
Okay, that makes sense. So once you know the structure, you can start to either experimentally or I guess probably even
In
maybe
silico.
maybe maybe computationally start to
Yeah.
figure out like, okay, we want to target it this way. Here's what we need to make, or here's what it needs to look like, and then start figuring out how to make those changes.
Yeah.
Do I have that
Yeah.
right?
Exactly.
Again, I'm not a biologist, so I may be completely wrong.
No, I think you are absolutely right
Okay.
here. And yeah, once we know that this is the substrate binding site and these are the residues which are interacting, so that gives a uh, really a great, basically site where you can actually, you know, um, you could jump in with different molecules and
Hmm.
different groups, chemical groups, to actually synthesize different stuff and see if it can actually bind or inhibit, because you do have we also have functional assays, so you can test those compounds in those functional assays as well to see if they're inhibitors or modulators, or like, or positive modulators or negative modulators of the activity. And that's how you can check out your, I mean, you can use those for the physiological processes to recover your disease state. So that's how it can be
Okay.
used. Yeah.
Okay. Interesting. All right. I have one more question about the second set of work that you showed with the two different assays on the excipients. Um,
Mhm.
is that not necessarily with those specific assays, but is that general methodology something that could be generalized? So, like, let's say you have a different, maybe not the glycoprotein. Yeah, I'm sorry.
P-glycoprotein.
Yeah,
So I'm trying to understand the question. Yeah.
okay. So can you generalize that method to other I guess other molecules? So like, if you want to understand like do this maybe the same set of excipients also inhibit other molecules? Is the method generalizable? I guess is what I'm asking.
Yeah, so yeah, you are right that method can be generalized. So on yeah, these are the methods that we used,
Hmm.
and yeah, method can be generalized by. I mean, if you want to change the transporter, So calcium AM assay can be used for MRP4
Mm-hmm.
as well as BCRP, as well as P-glycoprotein and other. So it depends upon your substrate, basically,
Okay.
like which substrate you are using and the detection method.
Okay. That makes sense.
Yeah,
That makes sense. Okay.
yeah,
But I mean, the way you did this was really cool because I mean, it seems I'm not going to say easy because I know science is never easy, but it seems like a straightforward method of
yeah.
sort of screening every a wide range of things relatively quickly and getting feedback and being able to interpret everything and compare apples to apples and really get a good feel for, okay, these ones inhibit, these ones don't do anything, and so then you can start to focus in on specific conditions that you're worried about and see whether you have any excipients that end up in that region. That's a really cool method you came up with.
Yeah, I think the first screen, the calcium screen was really really fast.
Hmm.
However, the other one, I mean, transfer assay, that was not very fast.
Okay.
But yeah, first one, calcium was a little faster because cells can grow very fast, and it was
Hmm.
96 well plate. But for transfer assay, it was 24 well plate, and also we had two chambers,
Hmm.
and then the MDCK cell line also took a little longer to grow, so that was
Okay.
time consuming. And then because we had two chambers, so pipetting, you know
Yeah.
pipetting goes like two ways, and you have lots of washing, and in between, measuring the transport, so you have lots of washing as well. So
Okay.
that's how you can actually, measurements, that takes time. And then 1 hour, 4 hours, 5 hours, I think that also takes
Oh
time. Yeah,
goodness. Yeah.
yeah,
This is also why I'm not in biology. I don't have the patience. That's really, it's like I said, it's really cool work that you've shown. I think that's all the questions we've got. So I will say thank you again, Ruchika, for, like I said, just really cool talk. This is super interesting work that you're doing. And we appreciate you sharing it with us.
yeah, thank you
Yeah.
for giving me this platform to present my work. Yeah.
Yeah, absolutely. Happy to do it. And, certainly feel free to come back. And for anybody that is watching this talk, Ruchika is on the job market. So you can see she does outstanding work. Uh, and say if you're looking for a membrane protein biologist, I think you found your person. So um, thank you again. And thank you to everybody for coming out. And we will be back next week at the same time. Hope to see you
Okay.
all then.
Yeah, thank you.